This video is an introduction to the nucleic acid topic, specifically thinking about the nucleotides found in DNA and RNA, as well as phosphorylated nucleotides like ATP. Nucleic acids are large molecules that are responsible for storing and transferring the genetic code. They're called nucleic acids because they were originally discovered inside the nucleus, although they're not exclusively found there. There are two types, DNA and RNA, although we can break RNA down into further subtypes. Now, you might already know that RNA is quite a bit smaller than DNA, and you might be thinking, why are we calling it a large molecule? But even quite a small RNA molecule is going to contain thousands of atoms. Both DNA and RNA are polymers because they are very long chains of repeating units called monomers, and the monomer of a nucleic acid is a nucleotide. Just as we've already seen with carbohydrates and with proteins, you're not going to have the exact same nucleotide repeated over and over. We are going to have variation, and that's what gives us the genetic code. Regardless of whether we're talking about DNA nucleotides or RNA nucleotides, the nucleotide has three basic parts to its structure. It contains a phosphate group, which contains phosphorus atoms and oxygen atoms. It contains a base, which is sometimes called a nitrogenous base, and in the centre of those two is a sugar, which is sometimes called a pentose sugar, because regardless of whether it's DNA or RNA, that sugar contains 5 carbon atoms. If we're talking about DNA, then the sugar is called deoxyribose, and if we're talking about RNA, the sugar is just called ribose. Let's look at DNA in a little bit more depth. You probably know that DNA stands for deoxyribonucleic acid, so DNA. And the name of this acid comes from the name of the sugar, so this is deoxyribose. You should also know that DNA can contain 4 different bases: adenine, guanine, cytosine, and thymine. We can group these bases into 2 pairs based on their size. So adenine and guanine are referred to as purines, and they contain 2 rings in their structure, whereas cytosine and thymine are pyrimidines, and they contain a single ring. It might be helpful to remember that pyrimidine has a Y in it, as do cytosine and thymine. Hopefully you're still with me, so quickly pause the video and write down the answers to these 3 questions. The 3 components of a nucleotide are the phosphate group, the pentose monosaccharide sugar, and a nitrogenous base. In DNA, that pentose sugar is called deoxyribose, and it's called a pentose because it contains 5 carbon atoms. When you studied the carbohydrate topic, and particularly when you thought about monosaccharides joining together to form disaccharides, you would have learned about the 1,4 glycosidic bond and also 1,6 glycosidic bonds if you're thinking about a branched polysaccharide. And you would have learned that in order to number those carbons, we start from the oxygen and then we move in a clockwise direction. And we use that exact same numbering system when we're talking about DNA nucleotides. And this is very important because when we read DNA, we're always reading it in the 5' to 3' direction. So that means starting from the carbon at the top and moving down to the carbon at the bottom on the left. And that carbon, you can see, has a hydroxy group on it. And that hydroxy group is really important when it comes to joining these nucleotides together to form a string of DNA. That carbon 3 has a hydroxy group on it, as does this phosphate group. And so, as we've talked about with other biological molecules, these are going to undergo a condensation reaction. This is going to lead to the loss of a water molecule and the formation of a phosphodiester bond. And this is the same regardless of whether we're talking about DNA or RNA. So pause the video, and can you describe how a polynucleotide is formed from its monomers? So we're thinking about multiple nucleotides being joined together by phosphodiester bonds, and this takes place as part of a condensation reaction. When describing the structure of DNA, you'll obviously have heard of the double helix, but you may also hear reference to anti-parallel strands. As we've said, it's really important that DNA is always read from the 5' to 3' direction. So if we number up all of our nucleotides, we can see here that the left-hand strand will be read from the top down to the bottom. But the right-hand strand will be read from the bottom up to the top. And this is what we mean when we talk about anti-parallel strands. DNA is a double-stranded molecule, and both of those strands terminate with a nitrogenous base. And those bases are matched up in what we call a complementary way. So that doesn't mean that they're exactly the same as each other, but it means that they always join together in a specific number of pairings. In each of these, we're always going to have one purine, which has 2 rings in its structure, and one pyrimidine, which has a single ring. And this is important because it means that the diameter of the DNA is always going to stay the same. So we have guanine joining with cytosine, where guanine is the purine and cytosine is the pyrimidine. And then thymine joining with adenine, where adenine is the purine and thymine is the pyrimidine. Now you can see here that there are a different number of lines between these, and this is because guanine and cytosine make 3 hydrogen bonds which hold them together, where adenine and thymine only make 2. So the combination of the number of bonds that the nucleotides make and the size of them makes sure that we're only ever having the correct nucleotides being matched together. Time for another progress check. Pause the video and see if you can answer these 2 questions. Well, we know that a nucleotide contains a sugar. So to start with, we're going to have carbon, hydrogen and oxygen. And then you've also got your nitrogenous base, so you have nitrogen, and you have the phosphate group, so you have phosphorus. Out of these 2 diagrams, only one of them could possibly be cytosine, because we know that cytosine is a pyrimidine, and pyrimidines only have a single ring. So therefore, the diagram on the left must be cytosine. An organism's DNA contains 17% adenine. Deduce the percentage of the other 3 nucleotides. Well, we know that adenine pairs equally with thymine, so therefore there must also be 17% of thymine. And then the remainder of the DNA must be split equally between cytosine and guanine. So therefore we're going to do 100 take away 34, which is 66%, and divide that by 2 to give us 33% of both cytosine and guanine. Now you need to pause the video and identify which of the nucleotides could contain cytosine. We know that cytosine is a pyrimidine, and that means it has a single ring structure. But also we know that cytosine always forms 3 hydrogen bonds to guanine. So therefore, the nucleotide containing cytosine could be this one or this one. As you know, DNA has a double helix structure. It's made of 2 strands of polynucleotides, in other words, polymers made from nucleotides, that are coiled into a right-handed helix. The 2 strands are held together by hydrogen bonds between the complementary base pairs, and the strands run anti-parallel to one another. So what that means is that if this strand is running from 5' down to 3', then the other strand will be running from 5 up to 3 in the opposite direction. RNA stands for ribonucleic acid, and unlike DNA, there are several different types. The 3 that you need to know about are mRNA or messenger RNA, tRNA or transfer RNA, and rRNA or ribosomal RNA. RNA provides the essential link between DNA and protein synthesis because it allows information to leave the nucleus. The DNA is a store of an organism's entire genetic code, so it's far, far too big to get out of the nucleus and interact with the ribosomes. And that's where RNA comes in, because it allows that information to be transcribed, which means copied out, and then translated, which means turned into amino acids. There are 3 main differences between RNA and DNA. The first one is the pentose sugar. So rather than deoxyribose, we just have ribose. The second difference is that while RNA may contain adenine, guanine and cytosine, thymine is replaced by uracil, which is another pyrimidine. The third difference is that whereas DNA is always double-stranded, RNA is often single-stranded. As the name suggests, when we compare deoxyribose to ribose, deoxyribose is missing an oxygen. So this is a DNA nucleotide with adenine attached and deoxyribose as the sugar, but if this was an RNA nucleotide, then the hydrogen that is down here on carbon 2 is replaced by a hydroxy group. So to summarise so far, DNA is double-stranded where RNA is mainly single-stranded. DNA contains deoxyribose where RNA contains ribose, and DNA has the bases guanine, thymine, cytosine and adenine, whereas RNA has guanine, uracil, cytosine and adenine. Pause the video and make sure you're confident answering each of these 4 questions. The 4 bases found in DNA are adenine, cytosine, guanine and thymine. In RNA, thymine is replaced by uracil. These nucleotides are joined together by a condensation reaction, and the bond between adjacent nucleotides in the same nucleic acid chain are phosphodiester bonds. You're not expected to be able to identify exactly all of the different nucleotides because you're not expected to know exactly what the bases look like, but you should be able to pause the video and identify whether this nucleotide is a DNA or an RNA nucleotide and whether it contains a purine or a pyrimidine base. The key here is that carbon 2 doesn't have a hydroxy group on it, so therefore this is going to need to be a DNA nucleotide. And then if we look at the base, we can see that we've got 2 rings, which is therefore going to make this a purine. So our correct answer is A. Finally, you need to know about a class of modified nucleotides called phosphorylated nucleotides, the most famous of which is ATP, which is sometimes referred to as the universal energy currency. ATP is based on a modified RNA nucleotide, and the base is always adenine. That's what the A in ATP stands for. But unlike most RNA nucleotides, this one has 3 inorganic phosphate groups, so ATP is adenine triphosphate. When the cell requires energy, this can be dephosphorylated. In other words, phosphate can be removed. And this process also releases energy. Thank you very much for watching, and I hope you found this a useful introduction to the nucleotide topic. If you did find this video useful, then don't forget to like, subscribe, comment and share the channel, and I'll be back soon with some more A-level biology.